Hello everyone, welcome to another video of this revision series. In this video, we will talk about basics of immune system. As a system, this type of immune system is not like our so-called system like gastrointestinal system or respiratory system, where we can actually trace the related organs one by one. This is quite diffusely present in our body. The organs that can be regarded as the parts of immune system are mainly bone marrow and thalamus, which confer various types of immunity to our body, and hence they can be called as the organs of the immune system. And the other organs that also contribute to this immunity are spleen, the largest lymphoid organ, the various types of lymph nodes present throughout our body, and tonsils to some extent. Next, if we consider any system into tissue level after organ level, the tissue that is responsible is mainly called as lymphoid tissue. And one of the examples like MALT present in the GIT, that is mucosa associated lymphoid tissue present in the duodenum or intestine. Next, the cellular level. The cells that are mainly contributing factor for the immunity of our body are leukocytes. All types of leukocytes in various way they confer immunity, and among them the most important cells are B and T lymphocytes, and hence the tissue which is related to the immunity is called as lymphoid tissue. Basically, they secrete more amount of lymphocytes and they provide immunity. The other types of cells that also play a major role in immunity are macrophage, natural killer cell, or null cell, because they lack any types of receptor recognizing molecules or memory cells, and they do not require cytokine to be activated, hence they are called null cell. The dendritic cell having dendrites like projection, and mast cell and plasma cell, the converted form of B cells, which secrete various types of immunoglobulins as a mechanism of humoral immunity. Now we will discuss the immunity and its classification. The term immunity can be defined as the resistance offered by the host against the microorganisms or any foreign substance. And immunity can be divided into two broad headings like innate immunity and acquired immunity. Innate immunity is the type of immunity that is the resistance to infection that an individual possesses from birth, and this type of immunity is very non-specific type, and hence it can be ensured within minutes. And the very basics of some innate immunity are like our skin, the largest organ of our body, which is present as a covering throughout our body surface from our scalp to our toes, and it confers the major or first degree line of defense to our body. Like it secretes sebum that contains some antimicrobial substance, and it kills the microbes from entering into our body. Next, the mucosal barrier. Mucosa is the inner lining of any important viscera, and they secrete various types of antimicrobial substance in the form of various secretions. Like in the tears, they contain lysozymes that easily destroy the peptidoglycan layer in the bacterial cell wall, or in the form of gastric juice, where HCl or lower pH can kill the bacterial protein. Some types of immunity may be conferred by the bile salt, which interfere with the bacterial cell membranes, and also cilia present in the lower respiratory tract propel the microbes outside from entering into the lower respiratory system. And also we know that there are present some normal flora in our intestine that are helpful for preventing the dangerous type of microbes. Also, the physiological barrier like our temperature and normal pH can prevent growth of microbes in our body. Next, the acquired immunity, that is the resistance to infection that an individual acquires during his lifetime. So this type of immunity requires some memory, and it is quite specific, more specific than the innate immunity, and it develops during the antigenic exposure. So the time required is quite more than innate immunity. It requires almost days to get action. And the immunity, this type of acquired immunity can be classified in various ways. One of the ways is like, according to the types, it is either active immunity or passive immunity. Active immunity means where the immune cells are directly activated to fight against the extracellular organisms or intracellular organisms. And passive immunity, where directly the weapon of the immune system are provided in various ways. They can be naturally given or artificially given. Naturally, like the classical examples of natural passive immunity is colostrum from the mother's milk, that contains the large amount of immunoglobulin A, which provide the baby to some extent certain immunity. And the artificial example like anti-tetanus serum or anti-snake venom, they directly provide the antibody against this tetanus toxin or snake venom toxin. Whereas the active immunity, here the immune cells like lymphocytes or other immune cells are directly activated with the exposure of some degree of antigens, either in the active form or in the passive form, like in the attenuated form. And these are the classical example is like vaccine, where the parts of the microbes or the specific antigen or the attenuated organism is given in the form of vaccine, and it provides immunity within months or years. Another classification according to the mode of action is the humoral immunity and cell-mediated immunity, which will be discussed in a later video. Humoral immunity is mainly by the B lymphocyte, which secretes various types of immunoglobulins after converting into plasma cells, and the humor term is given from the plasma mediated action of these B cells, and cell mediated, where directly the macrophage or dendritic cells or the natural killer cells secrete various types of enzymes and they kill the enzymes directly. Next, this is the primary and secondary antigen exposure with respect to our immune response. So the primary immune response, when the antigenic exposure occurs for the first time, then there is a lag phase followed by effector cells formation, and then the memory cell formation, and certain time there is a antibody surge, and this type of antibody are mainly IgM type. And when the same antigen is exposed for the second time, there is a certain high surge of immunity which is provided by IgG type, and this low titer of IgM is now replaced by high titer of IgG. So this is a curve obtained by the titer of immunoglobulins present in our plasma versus time curve, and the curve shows that in the primary antigen exposure there is low titer which is mainly IgM type, and in the secondary antigen exposure this low titer is replaced by high titer provided by immunoglobulin G. So this is all about, and obviously the lag phase is shortened here. The lag phase contains 4 to 7 days, whereas this is almost absent or very short. So this is all about the basics of immunity. Thank you guys for watching.